Chemokines and the role in immune system. Chemokines are molecules of the immune system that help in many functionalities of the immune system, and let us see how. So let us look at the learning outcomes of this session. Chemokines are a superfamily of cytokines that have a direct role in inducing migration of leukocytes, especially lymphocytes and dendritic cells. Members of the chemokine family are involved both in innate as well as adaptive immunity, with one of its hallmarks being chemotaxis. Chemokines are able to mediate activity through chemokine receptors, and these chemokine receptors are majorly the G protein coupled receptors. They have a profound importance in the genesis, homeostasis, and function of the immune system, and not only just to inflammation as it was earlier referred to. So, the chemokines per se are most known for the fact that they are able to function through chemokine receptors, which are G protein coupled receptors. So, as you can see over here, a chemokine, which has a typical structure of having a motif of antiparallel beta sheets and alpha helix, has three major domains. One domain is the one that is able to interact with the matrix, and the second domain is it allows the chemokine to bind to the receptor, and the third domain is once you have the second domain binding to the receptor, there is a conformational change within the receptor to allow the third domain to sit within the pocket in the receptor. Now, the word chemokine originates from cytokines. So, they are cytokines, but they have this ability to have the phenomenon of chemotaxis. So, they are basically attractants. They are chemoattractants. As mentioned, the cytokine receptor is a G protein coupled multipass transmembrane system. So we know that it traverses the membrane 7 times. So it is a 7 transmembrane receptor, and to its cytoplasmic domain is present the G protein. So when you have the chemokine binding to the chemokine receptor, the G protein gets activated and you would have a signal transduction thereof. So what has been observed is that generally chemokine-induced responses are quite rapid, but they are transient. That is because the G protein gets back again to its inactive form, and therefore it does not remain sensitized, and which is why you can see the response only transiently. Now, as mentioned, the chemokines are a folded structure, but what is very typical of the chemokines is that they have cysteine residues in a particular format. So you would have disulfide linkages present, and generally there are two disulfide linkages that are present, but the positioning of the cysteine determines what type of a chemokine it is. So when there is just one disulfide linkage, it is called as C chemokines. When there are two disulfide linkages and the CC are just adjacent to each other, they are called as CC chemokines. When the cysteines are separated by one particular amino acid residue within, then it is called as CXC chemokines. If there are more, then it becomes CXXC chemokines, and many of the chemokines are also associated with glycans. So you have a glycoprotein form also present. Now the general structure of the chemokine, the folded structure, it's very clearly seen that it has a beta pleated sheet and a alpha helix and two cysteine disulfide linkages. So this is how basically, and so you have this loop present over here and you have another loop that is present over here, and these play an important role in interacting with either the matrix or the chemokine receptor per se. So this is the basic structure of chemokines. Now, one of the major roles of chemokines is as an attractant. So as you can see over here, suppose you have a microbe that is present attached to a tissue, or you have a tissue that is damaged. So The microbe and the tissue, in response to the interaction, is able to release chemokines, and you can see that there is a gradient of chemokines formed. So here you will have more abundant, because this is where the chemokines are being secreted, but it can form a gradient, and therefore you can see that this is the range of chemotaxis. So if you have a leukocyte present over here, it will be attracted to this point because there is a larger concentration of chemokines present over here. So release of chemokines forms a gradient which recruits leukocytes to the tissues. So it is playing an important role in leukocyte migration just by being an attractant. Now, you would observe that even within the blood vessels, when the leukocytes have to go migrate, transmigrate through the endothelium into the tissue, that is through extravasation, then chemokines that are released by the endothelium actually enable the leukocyte to adhere to the basement membrane. There, the chemokine receptor interaction makes the leukocyte firmly bind to the basement membrane, and then you have the leukocyte migrating into the tissue. So again, you can see even within the blood vessels, you have the chemokine receptor interaction leading to leukocyte binding firmly to the endothelium, which then progresses to extravasation. It is also observed, however, this study is still on, that once the work of the leukocyte is over, then you can have another set of chemokines which actually allow the leukocytes to move back to the lymph node. So that is also something that has been observed, and therefore the leukocytes per se are moving in a cycle depending on the gradient of the chemoattractant present. Now, obviously, chemokines and receptors have been implicated in many functions, but the functions, as one can observe, has a lot to do with the immune system. So, for example, when one talks about inflammation, chemokine and its receptors have an important role to play. In homeostasis as well, for example, lymphocyte homing and dendritic cell trafficking, these are taken care of by the chemokines and the receptors. They have a role to play in cancer. Again, in this case also, the leukocytes infiltrate into the tumor because of the chemokine gradient that is formed. Then, of course, it gives rise to angiogenesis and angiostasis in immune responses against certain infections and also autoimmunity. So what has been observed is that chemokines can be inflammatory chemokines. In fact, when it was first understood to be part of immune system, then chemokines were only associated with inflammation. But over a period of time, it has been observed that chemokines, members of chemokines, are also involved in homeostasis, and so they are called as homeostasis chemokines. Moreover, chemokines modulate immune surveillance in tumorigenesis, metastasis, and immune response to therapy. So there is a lot of study that is relating chemokines to cancer per se. One of the major studies a major role of the chemokines that many studies have showed is that it plays an important role in dendritic cell trafficking. So here is an example of cancer research wherein you have the tumor, and the tumor cells, because they have molecular patterns of on their surface which enable the dendritic cells to recognize them as non-self. So the dendritic cell, precursor dendritic cells, because they have pattern recognition receptors, bind to the tumor cells, and in response to that they release several chemokines. Now these chemokines are attracting several other types of immune cells, plus they also ensure that the dendritic cells basically go to forming inactive dendritic sites, wherein the chemokines enable the antigen uptake, and because of this, what happens is that the chemokine environment enables the inactive dendritic cells to migrate to the lymph nodes. So you can see how the inactive dendritic cells slowly, gradually mature and then move to the lymph nodes where they become mature. And within the lymph nodes, we know that the dendritic cells are able to interact with CD4 plus T helper cells and CD8 plus T helper cells, thereby activating the T cells, which further then are able to initiate an adaptive immunity against the cancer cells itself. So, therefore, you can see how the dendritic cell trafficking is being influenced by the chemokines. So tumors are microbes that have the PAMPs, that is associated molecular patterns, pathogen associated molecular patterns can be recognized by dendritic cells, and the former attracts DCs, that is dendritic cells, by secreting chemokines. The chemokines help the migration of the inactive DCs to lymph nodes, where the inactive DCs can present antigens. And this in turn can activate naive T cells to enhance adaptive immunity. Now, the prevalent functions that are associated with chemokines are therefore definitely leukocyte chemotaxis, as we have seen in all the previous cases. So you can see how the leukocyte is able to walk towards, okay, or crawl towards or migrate towards a chemokine gradient. You can also have the leukocytes like, for example, basophils, eosinophils, mast cells, macrophages, etc., getting degranulated, and the mediators release are then responsible for a lot of inflammatory reactions. So in inflammation, therefore, chemokines have found a very important role, and they are also possible targets, therefore, for therapy. And of course, in extravasation, chemokine and the chemokine receptors enable arresting the rolling of the leukocytes. And once it is arrested and attached to the endothelium, then it can transmigrate into the tissue. So this is important. And another major area where it has found to be having a role is in angiogenesis or angiostasis. Plus, it is important to know that chemokines also help the immune cells to recognize a lot of foreign antigens. So let us make conclusions. Chemokines are small polypeptides with cysteine residues which are secreted to create a gradient that leads to phenomenon of chemotaxis, which in turn is able to recruit immune cells. Chemokine receptor family are of several types, with all of them coupled to G proteins and mediating a signal transduction. Chemokines can be inflammatory chemokines as well as homeostatic chemokines, contributing to both innate as well as adaptive immunity. Chemokines have been reportedly involved in DC trafficking and activation, in leukocyte migration and extravasation, in degranulation and release of mediators, and angiogenesis. So chemokines, as immunology's high impact factors, is intimately involved in migration of leukocytes, in inflammation, in adaptive immunity, and hence are potential therapeutic drug targets, especially for inflammatory diseases. Thank you.